This is a Cambridge. This paper covers almost all your topics, if not all the topics, arranged in the same order as the topics are in the syllabus. So the photograph shows the first question is on characteristics of living organisms and classification. So this question is actually on characteristics of life. So we are shown an animal here called a magray. A magray. 4 processes that occur in the magray are listed: excretion, growth, digestion, and respiration. Excretion is the removal of metabolic waste from the body. And these metabolic waste are produced where? They are produced in cells. Now, all living things have cells, whether prokaryotic cell or eukaryotic cell. No matter what the living thing is, it will have a cell. And in all cells, we have metabolic reactions that occur. The metabolic reactions that occur in cell produce metabolic waste that are removed during excretion. So all living things, whether bacteria, that is prokaryotes or eukaryotes, produce metabolic waste in their cells and remove metabolic waste. So excretion is a characteristic exhibited by all living things, as well as growth too, right? Unicellular organisms also grow. Their cells would increase in size. So bacteria grow. Amoeba, which is a unicellular eukaryote. It also grows. That's why they take in nutrients, right? Food nutrients, they feed. Nutrition. They take in food and they grow. So growth too is a general characteristic, but digestion is not a general characteristic. Digestion is typical of animals, fungi. Do plants digest? No. Plants make their food in chloroplast. And this food that is made in chloroplast is glucose. Animals ingest food. So as you can see, ingestion too is not a general ingestion, digestion, and ingestion. They all have to do with nutrition, but not all living things do this aspect of nutrition. But of course, nutrition is common to all living things. All living things need nutrients, food molecules to grow, for respiration, to make ATP, energy. But not all living things have their nutrition involve injection, digestion, and in fact, some animals don't even need to digest their food because they feed on already digested food, like tapeworm. Tapeworm does not digest food. Because tapeworm lives in the small intestine where we have what? Already digested food nutrients for tapeworm to absorb. So digestion is not common to all living things, even though nutrition is common to all living things, right? But the aspect of digestion called the aspect of nutrition called digestion is not common to all living things. Respiration. All living things need energy. All living things need energy. And the energy is made during respiration. So if you look at all the characteristics of life, right? You want to do Mr. Ninja. Mr. Ninja movement. Is it all living things that move? No. Reproduction. Is it all living things that reproduce? Yes. Nutrition. Is it all living things? Yes, all living things need food molecules to grow, to make energy during respiration. So all living things need energy, ATP. ATP is common to all living things, whether prokaryotes or eukaryotes. So respiration is typical of all. Now, but then, is it all living things that do aerobic respiration? No. No. But however, there's another type of respiration that does not involve oxygen, anaerobic respiration. So aerobic respiration, which involves oxygen, is not common to all living things. For instance, there are some bacteria that are called anaerobes or anaerobic bacteria. They produce energy via anaerobic respiration. They don't need oxygen to make energy. But so respiration is common to all living things, but not aerobic respiration. Just like nutrition too. Nutrition is common to all living things, but not ingestion, not photosynthesis. Not all living things can carry out photosynthesis. Sensitivity, which is irritability, right? Sensitivity is common to all living things. All living things respond to stimulus, dictate stimuli, and respond to it. All living things grow. All living things excrete metabolic waste. I hope this helps, right? So the answer here is what? Excretion, growth, respiration. So see. Now you need to understand your base pairing rules, right? Of course, when you see questions like this, that's what comes to your mind, your base pairing rules. But an aspect of DNA that we want to look at here is how genes, the study of genes can show what? Can show evolutionary relationship. Can show evolutionary relationship between organisms. For instance, when scientists decided to study the genome, they studied the genome of man, of chimpanzee, and gorilla. What did they discover when they studied the genome? What's genome? Genome is the total DNA in the body, total genes in the body, the entire genes in the body. They discovered that man and chimpanzee are more related. There's more relationship between man and chimpanzee than man and gorilla. Yes, man has 46 chromosomes, chimpanzee and gorilla have 48 chromosomes. But when they studied the base sequence, what do you study in the genome? The base sequence. The base sequence of genes, right? You study to look out for similarities. So we have species 1. You check out at how many points are they different. The more similar the bases they have, of course, the more related. So when you check species 1 and 2, first base the same, second base different, third base the same, different, the same, different, the same. So here we can see 3 base difference between them here. Now let's try to compare 1 and 3. The same, different. So we've seen another difference here. 1, the same, different, different. We are seeing 4 base differences. 4. Let's compare 1 and 4. 1 and 4. You see the same, different, the same, the same, the same, the same, the same, the same. Already we can see that the difference is more between species 1. When you compare species 1 and species 3, there are 4 base differences. The more different, the less related. So the species that are the least closely related will be species what? 1 and 3, because we see more base differences. A single-celled organism that has chloroplast, that has cell wall, nucleus, will belong to which kingdom? Of course, because it has chloroplast and it has nucleus, it can never be a prokaryote. It can never be a prokaryote. So that is out of the option. Now, because it has chloroplast, it cannot be an animal. Because it's also unicellular, single-celled, it can't be an animal. Of course, you may be thinking plant, right? Because it has cell wall, chloroplast, and nucleus. But it is unicellular. Plants are always what? Multicellular. But you're told that it is what? Unicellular. Unicellular prokaryote. Unicellular eukaryote are usually what? They are prototists. When you say multicellular eukaryote with cell wall and chloroplast, that's for plants. Multicellular eukaryote without chloroplast, but with cell wall. Then you're talking about fungi. Fungi don't have chloroplast, but they have cell wall and they are multicellular, mostly multicellular. Multicellular eukaryotes without chloroplast and cell wall, and you're talking about animals. Of course, unicellular prokaryote leaves you with no other but bacteria, right? The prokaryote. They are in their own, they are unicellular prokaryote. So here, unicellular, single-celled, chloroplast, cell wall, nucleus. That is obviously a eukaryote, not a prokaryote. It is not a plant because plants are multicellular. It is not a fungus because a fungus does not have, or fungi do not have chloroplast. And fungi are mostly not unicellular, they are multicellular. But prototists, single-celled, prototists can have chloroplast because they are eukaryotes. So we have some plant-like prototists, yes, that can have cell wall and have nucleus because they are eukaryotes. So the answer to this question is prototist, which could be, for instance, a prototist like Chlamydomonas. What is a leaf? Of course, a leaf is an organ. A leaf, like a root, like flower, they are organs in plant. Your leaf, your root, your flower, your stem are organs in plant. Your xylem, your phloem, your mesophyll, epidermis, these are tissues in plant. So while these are organs, these are tissues in plant. A student measured the length of a structure using a microscope, and the magnification was times 250. So your formula is magnification equals image size over actual size. Now the image size was 5 millimeters. What was the actual length? So you make this subject of formula. So your image size is from here 5 millimeters over your magnification 250. And when you do your calculation here, you have 250 in 5. That gives you 0 point. Then you have 0 here. 250 in 50 is impossible to, you add another 0, and you put a 0 here. 250 in 500, that's 2. So 0.2 millimeters. So you get 0.02 millimeters rather. Microscopic unit, that's micrometer. You multiply by 1,000. So 0.02 times 1,000. You move your decimal out, 1, 2, then you cancel 2 zeros here. You have 2. You just punch this into your calculator actually. They're doing all of this. So or this could have, we could have easily done this. You could have easily done this. 0.02 is 2 times 10 raised to the power minus 2 times 1,000 10 raised to the power 3. By the time you do your calculation, you're going to get 2 times 1 micrometer. Indices, right? You're going to get 2 times 1, 2 times 10 raised to the power 1 micrometer. That's the actual size in micrometer. Which row shows the condition when diffusion of oxygen into the blood is fastest. Of course, there's oxygen in the alveoli, and the oxygen in the alveoli diffuses into the blood in the capillary, pulmonary capillary, right? So there's blood in pulmonary capillary, blood in pulmonary capillary. Now, of course, the steeper, the higher the gradient between the concentration of oxygen here and the blood, the higher the rate of diffusion of oxygen into the blood, right? Also, the larger the surface area of the alveoli, the higher the rate of diffusion. So alveolar surface area should be high, concentration gradient should be high, then diffusion distance should be short. That is why the wall of alveoli is thin. Alveoli is thin wall so as to reduce the diffusion distance for oxygen between the alveoli and the blood. Also, when you look at capillary are what? They are one cell thick. The wall of capillary too is one cell thick, so that the diffusion distance can be short. So shorter diffusion path or diffusion distance, higher concentration gradient, higher surface area would increase the rate of diffusion, higher temperature from what you've learned before now. Okay, then we'll go to number 7. For number 7, uncooked potato cylinders of identical size were placed in 4 different liquids for 2 hours. You know, this is like an investigation, right? And the size of the potato cylinders was controlled in this particular investigation. Now, they are uncooked potato cylinders, right? Placed in 4 different liquids for 2 hours. So we should, in this experiment, be considering what the water potential is in the liquid and what the water potential is inside the cytoplasm of the cells of the potato. So water potential inside potato cells and water potential in the liquid that the potato is placed in. Now, if water potential is higher in the liquid than in the potato cells, then there will be movement of water by osmosis into the potato cells. But if water potential is lower in the liquid than in the potato cells, then there will be net movement of water into the liquid. Okay? The potato, as soon as water moves into it, the potato will increase in length, will become longer when water moves into it. When water moves out of the potato, the mass, the length will decrease. Of course, a solution that has high water potential is a dilute solution. Example, distilled water. Distilled water is one solution that has the lowest water potential. Distilled water has water potential of 0. Yeah. So that's the lowest, is the solution that you can think of to have the lowest water potential. So when you have salt solution, right? So you can have weak salt solution. The water potential of a weak salt solution will be low, but not lower than that of distilled water. Now, when you have a strong salt solution, the water potential of a strong salt solution, the water potential of a weak salt solution will be lower than that of distilled water. Even though it is high, right? Relatively higher than that of the strong salt solution. but it will still be lower than that of distilled water. So if you want to arrange in decreasing order of distilled of water potential, distilled water has the highest water potential, weak salt solution follows, then strong salt solution. So like pure water would have a very high water potential. A sugar solution that was less concentrated than the content of the potato cells. What this means is the sugar solution was less concentrated than the content of the potato cells, meaning that the water potential of this sugar solution is higher. It's like a weak, salt solution. So higher water potential than that of the potato cells. Obviously, pure water too has what? Higher water potential. In fact, you would expect that the water potential of pure water will be higher than that of the what? This sugar solution that was less concentrated than the content of the potato cells. A sugar solution that was more concentrated than the content of the potato cell, of course, that means that this sugar solution has lower water potential than the content of the potato cell. A sugar solution that was the same, that was the same concentration as the content of the potato cell, that means that they have the same water potential. It's just like what we have in the case of the red blood cell and the plasma. The red blood cell do not burst in plasma because there is no net movement of water between red blood cell and plasma. But in the first case, water moves into the potato cells. In the second case, net movement of water into the potato cells. In the third case, because the sugar solution was more concentrated than the potato cells, water will move out. Here, no net movement because there is no water potential gradient as they have the same concentration. No water potential gradient here, so there will be no osmosis here. So here, water osmosis here or net movement of water is into potato cells. So the potato cell will increase in length in 1 and 2 because water moves into the potato cells.
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the Edexcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about and the fee is really quite affordable. It's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable and you get the very best of service. We also have for the E-levels as well, and also for the Edexcel. So we have for the Cambridge E-levels and Edexcel E-levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E-level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time, even physics, for E-level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at ajiro@gmail.com and they will reply to you.
enzymes. Of course, enzymes are proteins made of amino acids. They are not carbohydrates. They are proteins. And they act as what? Biological catalyst, not as substrate. An enzyme is a protein that has an active site that the substrate can bind to. The substrate of an enzyme can be a carbohydrate, can be a protein again, can be a lipid, can even be nucleic acid. But the protein, the enzyme rather, itself is always a what? Is always a protein. The substrate can be any of those biological molecules. So a student investigated how the rate of photosynthesis is affected by carbon dioxide concentration, temperature, and light intensity. Of course, for carbon dioxide concentration and light intensity, the enzymes do not get denatured. But for temperature, for factors like pH and temperature, they always denature enzyme when they become extreme. So at extreme temperature, you can see that here, the reason why the rate of photosynthesis is dropping back to zero, because the activity of the enzyme of photosynthesis is going back to zero because it's getting what? The enzyme is denatured. The enzyme has lost its active site. The shape of the active site has changed. At this point too, whenever you see this in the graph, this is the optimum temperature where the activity of the enzyme is the highest. But usually at the start of the curve like this, you always say that the independent variable here, which is always on the x-axis, is the limiting factor at this point, at this slope. But here, no longer the limiting factor. Something else is limiting the rate of photosynthesis here now, not light intensity. It could be carbon dioxide concentration, it could be temperature, but not light intensity. Because as you continue to increase light intensity, see, you're increasing light intensity. Here, we have higher light intensity. And the rate of photosynthesis refuses to increase. That means it's not light intensity that is limiting the rate of photosynthesis any longer. But here, when you increase light intensity, what happens to the rate of photosynthesis? It increases. So carbon dioxide concentration is a limiting factor at 1. Let's check 1. Is carbon dioxide concentration a limiting factor at 1? Of course, yes, it is. From what I just explained, that when you look at the slope here, the independent variable is the limiting factor. It's the one that has been slowing down the rate. So as we increase it, the rate increases. So carbon dioxide concentration is the limiting factor at 1. No enzymes are denatured at 3. That's not correct. Enzymes are denatured at 3, temperature. Light intensity is a limiting factor at 4. That's not correct. Light intensity is only a limiting factor at this point, and it's not numbered. Temperature is a limiting factor at 2. That's not correct. At this point, temperature is optimum. There's enough temperature that makes the enzyme to be at its best, to work at its best. There is optimum temperature at that point. So the answer to this question is option A. The diagram shows us a cross-section of the part of the leaf. So don't forget, palisade, closely packed together, cells that are closely packed together and contain numerous chloroplasts. Palisade, mesophyll. And this place where we have S species, spongy, mesophyll. Of course, this is what this place here is the vascular bundle. So we have xylem and phloem. This is xylem here. And right within it, you have phloem. So that's the vascular bundle. You have your upper epidermis, and you have your lower epidermis, where you find gut cells. So these are gut cells. Gut cells control the opening and closing of stoma. So this is stoma. One stoma, more than one stomata. I hope you also know that gut cells contain chloroplasts, right? But of course, not as many as you have in spongy, and of course, as you have in palisade. Palisade have the highest number of chloroplasts, followed by spongy, then gut cells. Spongy mesophyll, palisade mesophyll. So S is your palisade mesophyll. Number 11. In which part of the digestive system does physical digestion occur? Of course, when we talk about physical digestion has to do with action of teeth, grinding action, the grinding action of teeth. Physical digestion is also mechanical digestion. It does not involve enzymes, right? It's not chemical, it's physical. So the grinding action of teeth, as well as the churning of food by stomach wall. So the stomach wall is muscular and rough. So it presses on food and that helps to break down the food by the process called churning. The essence of physical digestion is to increase surface area for enzymic attack. The surface area of the food, after broken down into particles by physical digestion, is large enough for enzymes to now attack the molecules in the food and break them down by hydrolysis. So what the enzymes will do will be hydrolysis, right? Break down starch eventually into glucose, break down proteins. That's for enzymes now. Enzyme will break down the proteins into amino acids, lipids into fatty acids and glycerol. So the mouth and the stomach, physical digestion. Chemical digestion takes place in the mouth, the stomach, the small intestine, where we have final absorption of food, the small intestine, where food is finally absorbed by villi. The table shows the substrate and end product in 4 enzyme-controlled chemical reactions. So which reaction was controlled by lipase? Of course, we know lipase chemically digests lipid. So the substrate is lipid, just as I mentioned not long ago. Lipase is a protein. So lipid gets into the active site of lipase. So lipase breaks it down into what? Into glycerol and what? Fatty acids. So what's the substrate? Which of this is a lipid? Oil. The microvilli in the small intestine. Microvilli are found on the surface of cells that line the epithelium of the small intestine. When you look at the epithelium of the small intestine, which form villi, on the surface you will find these microvilli cells that have microvilli like this. Both villi and microvilli will increase surface area for absorption of food nutrients. Movement of mucus, that is carried out by what? Ciliated cells in the trachea. Ciliated cells are different from cilia, are different from microvilli, right? Cilia are structures on their own, but microvilli are what? They are foldings of the cell membrane. These ones move mucus to the throat in the respiratory tract. Which row shows the features of xylem vessels? Xylem vessels are made up of dead cells that are arranged end to end, and the wall of the vessel contains a thick substance called lignin. So the wall of xylem vessel is thick. Why? Because it has what? Lignin. This is also responsible for why xylem does not just transport, but also a what? But also provides support. So lignin is the reason why xylem is a supporting tissue and not just a transport tissue. So the wall is thick, but they are dead cells. So there can be mitochondria. The cells that make up, the xylem cells that make up the xylem vessel are dead cells, empty lumen. So thick, but no mitochondria, no cytoplasm, nothing, just empty lumen like this. Okay, so this is an experiment to investigate the effect of humidity on transpiration. And so the students set up two sets of apparatus with identical size plant of the same species. So there is oil air to prevent water from evaporating from the tube. So the plant will absorb water, and the water will be lost in form of water vapor. But here, there's a plastic bag that is covering it. So the water vapor stays where? It stays within the plastic bag, increasing what? Humidity. So this increases humidity. Humidity has to do with amount of water vapor. So humidity is increased here. And because humidity is increased here, the water vapor gradient between the leaf and the outside of the leaf, the gradient is going to be very low. But here, the plant is exposed, nothing covering. So here, humidity is low. The amount of water vapor here is low, especially when temperature is high, wind speed is high, light intensity is high. Of course, humidity will be low. So the gradient, the water vapor gradient between the inside of the leaf and the outside will be steep. So inside leaf, outside leaf, the gradient is steep because of the low humidity. So more water vapor will diffuse out, be lost from here. But this one, there will be less water vapor diffusing out of these leaves. So if you look at that, from the experiment, what do we observe? As water diffuses out of the leaf, the mass of the plant will reduce. So when you look at plant Y, where there is less transpiration, the change in the mass of the leaf is going to be small. Compared to X, where a lot of water vapor diffuses out. So you're saying that there is more change in the mass. There's more change in the mass in X than in Y. And the reason is that the rate of transpiration in X is higher than the rate of transpiration in Y. So in other words, you can say transpiration in Y was lower than in X. And the plastic bag trapped what? The plastic bag trapped water vapor deep, right? Trapped water vapor. And so we had more water vapor inside the bag, making humidity high and transpiration low. It is low humidity that increases transpiration. But when humidity is high, transpiration will be low. Low humidity, high rate of transpiration. High humidity, low rate of transpiration. So transpiration in tube Y was lower than in X because the plastic bag increased humidity. So let's move to the next question. Now for the next question, what is an advantage of double circulatory system? Yeah, like we have in mammals, right? The reason why mammals have double circulatory system or exhibit double circulatory, have double circulatory system or exhibit double circulation is because mammals have a four-chambered heart. Four-chambered heart. The oxygenated blood moves into the left atrium. Sorry. The oxygenated blood from body cells move into the right atrium. Then from the right atrium, the oxygenated blood goes to the right ventricle. Then the pulmonary artery takes the blood to the lungs to get oxygen. Now the blood comes back from the lungs, being carried by the pulmonary vein. It's carried to the left atrium. Then the blood flows to the left ventricle. Then the aorta will take the blood from the left ventricle to the body. The wall of the left ventricle is so thick that the pressure there is so high, making the blood that is going to the body to be of higher pressure. This will allow blood to reach body cells faster. Blood can flow from the left ventricle to as far as what? As the toes. So blood will reach the toes faster because of the high pressure that is generated in the thick left ventricle. But you know, in fish do not have four chambers. Fish have heart with what? Two chambers. So the blood comes to the heart, enters into the heart through the atrium, then the blood to the blood. So an advantage of a double circulatory system is that oxygen is pumped to body cells. Oxygen is supplied to body cells by high pressure. You know, for fish, blood flows from the gills to the body cells under low pressure because the heart does not have four chambers to receive blood from the gills. But the heart of mammals has four chambers. There's a left atrium to receive blood from the lungs, and there's a left ventricle to pump blood to body at high pressure. But in fish, there's only one atrium and one ventricle. So there is no way blood will pass through the heart twice. Which structure in the heart separates oxygenated and deoxygenated blood? Of course, when you look at the heart, the structure of the heart, the mammalian heart, you would see the septum. The septum separates the left from the right part of the heart. Right, they have the septum. That's the septum. Of course, between the chambers of the heart, we have the valves, right? Between the chambers of the heart, we have what is called the atrioventricular valves between the chambers of the heart. The vein, a vein has a large lumen, and the wall of the vein is less thick. Arteries have smaller lumen but thicker walls because of the high pressure of blood in arteries. To be able to withstand the high pressure, they have thicker walls but smaller lumen. But veins don't conduct blood of high pressure, so they are large lumen with thin walls. So when you compare the relative thickness of the wall to total diameter of the blood vessel, for veins, it is thin. The diameter of the lumen, as you can see, is large. So the wall is thin, the diameter is large. Of course, to prevent blood from flowing backwards, veins have what? They have valves. Because the pressure of blood in artery is high, arteries don't need valves. But veins, because of the low pressure of blood, so that the blood does not flow backward, veins have valves. So the answer to this is D. 19, immunity. Of course, we can gain two types of immunity. We can gain active immunity, and we can gain passive immunity. So how do we gain active immunity? Active immunity can be gained through vaccination, when vaccines or antigens, right, the weakened pathogen when it is injected into the body. We can also gain active immunity through what? Through infection. So in both cases, the lymphocyte will produce antibodies. So in both cases, in both ways that we can acquire or gain active immunity, whether vaccination or infection, lymphocytes produce antibodies and also produce memory cells. Antibodies will destroy pathogens. Now, memory cells would remember pathogens when pathogens come into the body again. So memory cells give long-term immunity. Yeah, long-term immunity. Now, but when it comes to passive immunity, in the case of passive immunity, lymphocytes do not produce antibodies, and memory cells. So passive immunity is short-term, as there are no memory cells. So how do we get passive immunity? Passive immunity occurs or is gained by getting antibodies from external sources. So a baby that gets antibodies from the milk, the breast milk, or a patient that is injected with antibodies, we get passive immunity. So in passive immunity, the lymphocyte of the person
a lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the EdExcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have um, the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about, and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable, and you get the very best of service. We also have for the E levels as well, and also for the EdXL. So we have for the Cambridge E levels and EdXL E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for E level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at ajiro@gmail.com, and they'll reply to you.
Definitely here, number 19. From what have been explained so far, it means that the transfer of antibodies from mother to baby in breast milk is an example of passive immunity, as the body of the baby does not produce antibodies. The antibodies are the antibodies of the mother. Passive immunity results in long-term immunity because of the production of memory cells. No, that's not correct. Immunity that involves lymphocytes producing memory cells and antibodies. So this is not correct. This is correct. Active immunity is gained after vaccination with antigens. This is correct, right? So the answer here is 1 and 3 only. That's 19. 1 and 3 only. So let's review this. They say which statements are correct? Okay, 1 is correct. Passive immunity results in long-term. Yeah, 1 and 3 only correct. Let's move to question 20. And for 20, the table shows the breathing rate and the average total volume of air breathed by a student when exercising for 20 minutes. So in the first 5 minutes, the breathing rate is 12 breaths per minute. Started with 12 breaths per minute. After 5 minutes, it's still taking 12 breaths per minute. After 10 minutes, it's still taking in 12 breaths per minute. But when you check the volume of air that is taken in, right, even though it's taking 12 breaths per minute, but more air, right, is being taken in. Breath per minute, breath is just talking about, of course, the uh, the heartbeat, the heart pulse. Talking about is inhalation and exhalation. But this talks about the volume of air that it takes in when it breathes. So as you can see, it's taking in more air as time increases. So between 0 and 10 minutes, the depth of breathing increases because we can see between 0 and 10 minutes, the depth of breathing increases because more air is taken in. That's the answer. The chemical formula of glucose. Same for fructose. The chemical formula for sucrose. So this is for disaccharide. This is for monosaccharide. So after a race, athletes experience oxygen debt. What is oxygen debt? Oxygen debt is volume of oxygen needed to break down lactic acid. To break down lactic acid during aerobic respiration. So when animals carry out anaerobic respiration, lactic acid forms in the muscles. And the lactic acid, after the exercise, as the lactic acid is building up during the exercise, right? Lactic acid will go from the muscle into the blood, and the blood will transport the lactic acid to the liver, where oxygen can be taken in. So extra oxygen, that extra oxygen that is needed to break down lactic acid during aerobic respiration. So when the lactic acid gets to the liver, it has to be broken down by aerobic respiration. So the extra oxygen is supplied by faster breathing after the exercise. So when you carry out an exercise, a rigorous exercise, or you run a race, you discover after the exercise, you breathe heavily. At that time, you're trying to pay the oxygen debt. You're trying to take in the extra oxygen that is needed to break down any lactic acid that must have been formed in the muscle during an aerobic respiration. So what simply happens here is the aerobic respiration of lactic acid. So oxygen is needed for aerobic respiration of lactic acid, right? So extra oxygen that is needed is called the oxygen debt. Okay, another function of the liver, apart from aerobic respiration of lactic acid, another function of the liver is production of what? Production of urea. The liver produces urea by what? Deamination of what? Excess amino acids. So when we eat excess protein, we have excess amino acids in our body. Our body cannot store excess amino acids the way it stores excess fat and the way it stores excess glucose. So the liver has to break down the excess amino acid by the process called deamination. And in the process, urea is made. So the deamination of excess amino acid leads to what? The production of urea. The diagram shows a vertical section through a part of a human eye. A fly is coming nearer to the eye. The eye begins to focus the image of the fly on the retina. Okay, so the fly is coming closer. Now, what happens when we see close objects? Now, whenever we see close objects, our lens becomes thicker, more convex whenever we see close objects. This is just the normal size of the lens, right? The normal shape of the lens. So when we are seeing a close object, the lens is thick. The suspension ligaments are relaxed or slack. They are not working. but the ciliary muscle will contract, right? Will contract when you see a close object. So as the fly comes closer to the eye, the lens becomes fatter or thicker or more convex, right? The normal, right? Lens usually becomes thin when you are seeing a distant object. So if this fly should go back, if it's distant, this lens will be flat or thin like this, because at that time the suspensory ligament will contract, pull or stretch the lens, right, so that it can see distant object. At that time the ciliary muscle will be relaxed, but suspensory ligament will be contracted. When you are seeing distant object, your lens will be what? Will be thin. So because the fly is coming closer to the eye, the suspensory ligaments are relaxed or they are slacker. They are not tighter. They are going to be slacker on the lens so that the lens can assume its normal convex shape. So the lens, which is hard, will be fatter. So it can be A or B. Is either B, it can be A or D rather. Is either B or C. So the lens is fatter and the suspensory ligament will be slacker, but the ciliary muscle will contract. So the answer is C. Glucagon is a hormone. Glucagon is the hormone that increases blood glucose level. Another hormone that increases blood glucose level, adrenaline. So glucagon is produced by the pancreas. Right? Glucagon is produced by the pancreas. Adrenaline is produced by the adrenal gland. A young seedling is growing underground. For a young seedling that is growing underground, right, the shoot, the plumo, the shoot, is trying to come out of the soil. Yes, it's trying to come out of the soil. It's trying to come out of the soil. The root is trying to grow down the soil. So when you look at it, the force of gravity acts downward. Force of gravity. The force of gravity acts downward, right? And up here you have what? You have light, right? So if you look at the shoot is trying to respond to light, and gravity is growing against gravity. So the shoot is exhibiting phototropism and gravitropism, because yes, it's responding to gravity, growing against gravity, growing towards light. The root, as far as the root is concerned, the root is growing in line of gravity, in line with gravity, right? So the root exhibits gravitropism by growing in line with gravity. The shoot is growing against gravity. So both root and shoot are responding to gravity. This one is growing against gravity. This one is growing in line with gravity. The root is under the soil. There is no light there. So the root is not responding to light. That's phototropism. But the shoot, okay, coming. The shoot grows towards the surface of the ground. Okay, the root grows away from the surface of the ground. Yeah. So if you look at this, actually, this it didn't bring in light, right? Light will only come in when we are talking about placing the plant in a environment where it's getting light from one direction. That is when the response is phototropism, right? So we say the shoot is bending towards the source of light. The root is bending away from the source of light. That is a case of phototropism. But this case that we are talking about, growing upwards and downwards, growing towards the ground and growing away from the surface of the ground, that's gravity. So the stimulus here is just gravity, please, not light. The stimulus that is being responded to is gravity and not light. So that's why it is root, yes. Root is growing in line with gravity. Shoot is growing against. So both of them are responding to gravity. So while one is showing negative response, that's the shoot. The root is showing what? Positive response. So this case is just a case of gravitropism, response to gravity. So A is the answer. 27. Clostridium difficile is a species of bacterium that is resistant to many antibiotics. Of course, you know this is the scientific name, the genus, and the species. From 2002 to 2006, the number of people in one country infected with this particular species of bacterium increased from 1,300 to 6,200. Now we are asked to calculate the percentage increase. How do you calculate percentage increase? Increase over original times what? 100 over 1. The increase here between 1,300 and 6,200, you're going to find a difference. And the difference is 4,900. So increase is 4,900. That's the difference between the two of them. And the original is 1,300, not 6,200. The original, it increased from 1,300, the original, to 6,200. So increase over original 1,300 times 100 over 1. If you do your calculation, that gives you what? 377%. Asexual reproduction involves one parent, no fertilization, no production of gametes, no meiosis, no genetic variation in the offspring. So genetic variety in the offspring, no. Fusion of gametes, no. No fertilization in asexual reproduction. So the answer here is the two features given here are of sexual reproduction. Now for cross-pollination involves transfer of pollen grains from the anther, right, of one plant to the stigma of another plant. Pollen grains are produced in the anther and they can be carried by wind or by agent of pollination like birds too. Birds, winds, insects are agent of pollination in the case of cross-pollination, carrying pollen grains from the anther of one plant to the stigma of another plant. It is self-pollination when it is carried from the anther to the stigma of the same plant. When we talk about menstruation is the shedding of the lining of the uterus. Ovulation is the release of egg from an ovary, which usually occurs when ovulation usually occurs when in the middle of the menstrual cycle, around day 14, and is stimulated by luteinizing hormone. At the beginning of the menstrual cycle, that is when menstruation happens. In the middle, that is when ovulation takes place. So ovulation is the release of egg from the ovary into the oviduct, where fertilization takes place. Menstruation is the shedding of the lining of the uterus. Of course, when you look at this diagram here for 31, if you look at the inheritance of the disease, you will discover that the disease is seen in this offspring here and in this offspring here, meaning that the allele that causes the disease is dominant, is the dominant allele. If the allele were to be recessive, right, this will need to be heterozygous dominant to see offspring with the disease. Because if this was to be homozygous dominant and this genetic condition was recessive, you will not see any offspring with the disease. But because the condition is dominant, we see offspring with the disease, right? Yeah. So the allele for the condition is caused by a dominant allele. That is what the pedigree diagram is showing us. What is the way in which alleles are formed? So a change in the base sequence of a gene leads to formation of new alleles. So if, for instance, if we used to have this particular base sequence, CG, A, T, T, G, A, C, C, A, G. Now, this is a particular base sequence. It could be a specific gene. So what does mutation do? Mutation can just cause a substitution of this particular base here. The base here, T, could just be substituted to what? To A. That's a mutation. That's what happens during mutation. And what has just happened? A new form of the gene has just been what? Introduced. So what do we call a new form of a gene? Or what do we call forms of a gene? Alleles. It is actually mutation that produces or forms new alleles of genes, not natural selection, not sexual reproduction or artificial selection. Sexual reproduction only does what? Reshuffles existing alleles of a gene, making offspring to have different combinations of the alleles of a gene. And what does that cause in sexual reproduction? Variation. So variation is caused by sexual reproduction. For instance, meiosis, random mating, random fertilization during sexual reproduction can cause variation by reshuffling existing alleles by recombining existing alleles. But for a new allele to be introduced, for instance, we used to have A and S as the only allele for what? Hemoglobin gene. But today we have a new allele, C. This new allele is appearing as a result of what? Mutation. And that's going to introduce a new variation. Artificial selection differs from natural selection. You know, artificial selection always involves human intervention. It is human that will unpick the characteristics that is to be favored, not the environment, not competition between organisms in the environment. So there's usually no competition. It is just the farmer, the breeder, that unpicks his favorite traits and then start breeding organisms with this favorite trait over a long period of time until he gets a uniform population. So if you look at it, artificial selection does not involve organisms competing for resources. But in natural selection, there is variation, there is competition, right? There is survival of the fittest. There is production of large number of offspring. This is one of the causes of competition. Then the offsprings with better variations are better adapted to the environment, so they will outcompete the weaker organisms. So they are the fittest, they survive. That does not happen in the case of artificial selection. Artificial selection just involves man and picking the breeder and picking his favorite, right, based on the desired traits. Now, when you look at this is a cactus plant, a desert plant. Desert plants usually have extensive root system so that they can absorb water from the soil, reach the water table. So they don't have restricted root system. They have extensive root system. And of course, they have this waxy layer, waxy cuticle, thick one, to reduce the rate of water loss by transpiration, evapotranspiration. '35. Which row describe the energy flow into and through a food chain that starts with a plant? The ultimate source of energy in the ecosystem is the sun. So plants absorb sunlight energy, light energy. The chlorophyll in plants absorb light energy. And in the chloroplast, what happened? The light energy is used to make glucose. What's the energy in glucose? Chemical energy. So when you look at photosynthesis, photosynthesis converts light energy to chemical energy. Now, it is that chemical energy that will be transmitted or transferred in the food chain. So when an herbivore comes to eat this plant, it's trying to get the what? It's trying to get the chemical energy. It gets the chemical energy. The herbivore gets the chemical energy in the glucose, right? Then the carnivore comes to eat the herbivore. What is he also getting? He's getting the chemical energy. So it is light energy first, then chemical energy is being transferred between organisms in the food chain. And that is what? Option light to chemical. Option C. Chemical energy in food, right? Now, in the food chain, zebras are large mammals that eat grass. The zebras have many insects living on them, which suck their blood. Birds eat the insect. Now, when you look at this food chain, what do you see? You observe that there are grasses eaten by zebras. Then insects feed on zebras. Then birds feed on insects. That's the way the food chain will go. Grass to zebra, zebra to insects, and insects to birds. So which row identify the types of pyramid for this food chain? Pyramid of energy. Now, remember that for pyramid of energy, energy always flows from producers to secondary, to primary consumer, to secondary consumer, to tertiary consumer, to decomposers. Although it's not good for the food chain to be longer, because the longer the food chain, the more energy that will be lost, right, at every trophic level. The less energy that the final organism will get. So shorter food chains are more what? Energy efficient. So if you have a food chain with just 3 organisms, it's more energy efficient than when you have 5 or 4 organisms. So here, energy is the highest at this trophic level. Energy is the highest at this trophic level. followed by this trophic level, followed by this trophic level, followed by this trophic level. This trophic level grasses, they get the most energy from sunlight. Zebra that will eat grasses will get more energy from grasses. Like, they do not get the whole energy in grasses because some energy will be lost in form of heat from the body of the grasses. And they do not eat the whole grasses, right? They may not eat the whole grasses. So they don't get, so imagine that we have 1,000 kilojoules of energy in grasses. What zebra will get after eating grasses may just be 800 kilojoules of energy, because energy is always lost at every trophic level in form of heat and other forms. So by the time the insect too, they suck the blood of the zebra, they are just sucking the blood of the zebra, the amount of energy they get will be reduced, maybe say 500 kilojoules. By the time birds too are eating insects, they are getting small amount of energy. They are not getting the whole 500 kilojoules, they are getting like say 300 kilojoules of energy. So if you look at the pyramid of energy, the pyramid always starts from the base with the producer, the primary consumer, the secondary consumer, right, and the tertiary consumer. So the long bar here shows that energy, there's more energy here, which is 1,000. There's more energy here, which is what, 800. There's more energy here than this place, which is 500. And this place is just 300. So as you go in the food chain, energy is lost. So this represents pyramid of energy. But how about pyramid of numbers? Pyramid of numbers now take into consideration the population, the number of the organisms, you know, that you have, the different. So let's start again from the base. Let's start with grasses. Of course, we have many grasses. Yes, we have so many grasses. Grasses are many if you count them in a population. But look at insects. If you look at insects, you will discover that there are actually insects. There are so many insects. Insects are very successful organisms. If you have a population of insects, you can count and keep counting. But what about zebra? Look at zebra. Zebras are big. They are large mammals. And due to this large size, you will not count so many of zebra. That's why if you look here, see the number for zebra will be small. Number. Zebra cannot be more than grasses. No, it's not possible. Zebra cannot be more than grasses. So this is wrong. This is out of it. This one is also saying insect to be the smallest. No. Insects cannot be smaller than birds in terms of number. There are more insects. If you have a population of insects, you will count more. You have a higher population of insects than a higher number of. So this is what goes for the pyramid of. Organic material like protein, like urea, converted to ammonium ions. Then nitrate ion. We have nitrifying bacteria convert ammonium ions to nitrate. Nitrifying bacteria like Nitrosomonas, Nitrobacter, Nitrosomonas nitrobacter, we convert ammonium ions to nitrate ions. Nitrate ions, of course, are used in making what? Amino acids, right? by plant. So plant will absorb nitrate ion. And this process is called nitrification. For nitrate ions to ammonia to nitrogen gas, that process is called denitrification. This one removes nitrogen from the soil. Nitrogen gas to nitrate ion, that's called what? Nitrogen fixation. So the process labeled X is nitrification. The graph shows the number of water fleas in a newly created pond. Now what happens? The number was low but constant. Then the number started increasing. This could be due to increase in birth rate, right, of the water fleas, or less predators. Yes, this will be due to increase in birth rate and less predators, or less death rate. Increased birth rate, less predator, more food, less or no predators, more food, increased birth rate, no disease. There's no disease. Decreased death rate. That's this place, right? This place. At this point, we could say death rate is equal to birth rate. Well, what happened to the decline? Of course, you can see here that the number is coming down, which means here death rate is higher than birth rate. Or there are predators. So there's no place that says that death rate is higher than birth rate. This place could also be that food supply reduced, so they are starving to death. So now there's no option that says decrease in food supply. There's no option that says increase in death rate. But there's an option that says addition of what? Predators, which feed on water fleas. So increase in predators, death rate. That's the answer. 39. Yeah. When managing fish stocks in a pond as a sustainable resource means you're trying to conserve, right, fish stocks so that there can be fish stocks in the future, so that all the fish stocks will not be exhausted. So we are trying to manage to ensure sustainability for future use. So what's the reason for harvesting larger fish, a particular species, and not harvesting smaller ones? Larger fish are easier to harvest. That's not correct. Larger fish sell at a higher price. Now, it's not because of the sustainable resource here. It's not about the price. It's about ensuring that the resource is sustainable and be available for the future. Now, we're taking out the larger fish, because the larger fish can breed young ones for future. So when managing small fish stocks as a sustainable resource, the reason for harvesting only the larger fish of a particular species It's because the larger fish can breed. Produce young ones for future use. The smaller fish are too young to breed. So, and the larger ones are picked. So why are bacteria useful in biotechnology? The reason is they can produce complex molecules like proteins, for instance, like insulin, right? Like blood clotting protein. And of course, they produce rapidly. Other reasons is the fact that they also use what? They use the same genetic codes. Yes, GC80 as other organisms. The genetic code in humans is the same as in bacteria, it's the same as in plants. Another reason is that they have plasmids, right? They contain plasmids. These are the reasons why bacteria are useful in biotechnology. So these two will not go. Yes, they make complex molecules like proteins. They reproduce rapidly. They have plasmids. They use the same genetic codes, as other organisms. So the answer here is. So that brings us to the end of the video, the correction.